Thank you.
You're welcome. The vestibule, the inner ear, vestibular apparatus and equilibrium. Okay, this is the vestibular apparatus is what's going to be allowing us to interpret movement, rotational, linear. Okay, are you going around in a circle? Are you standing still and going up or down, or standing still and on an escalator or some kind of moving walkway? All right, this is the part of the ear that's involved with that. Specifically, it's involving the semicircular canals, anterior, posterior, and lateral, and the X, Y, and the Z axis. And those are going to be associated with rotational movement. And it's done with some specific structures found here within the ampulla. Our otolithic membrane, our otolithic structures are going to be here within this vestibule right here. And this vestibule consists of a couple small structures called the utricle and the sacculae. And inside those regions are going to allow us to interpret linear movement, linear acceleration, okay? Again, this is more of standing still and your body's in motion, whereas now I'm in motion, okay? This is dynamic, all right? This is dynamic equilibrium. Dynamic, E-Q-U-I-L-I-B-R-I-U-M, equilibrium, okay? And this is more static, okay? You're not necessarily in motion on this one. That's all that means, okay? It's static. I don't want to spell equilibrium again. Okay? When it comes to this dynamic equilibrium, it's going to involve fluid movement, all right? Fluid movement within the tube pushes structures within the ampulla there, and I'm going to interpret that as going left or right. And that's actually learned, right? You learn when you go this way, you're looking to the right. You learn your left from your right, right? So it's not that your brain interprets left and right, right? It just interprets motion in this direction and in this direction, right? You determine left or right. You learn left or right. The utricle and the sacculae, it has actually this otolithic structure. It's a jelly with pebbles in it. Okay, otolith means pebble, stone. All right. And these otolithic pebbles that you have there are embedded in that jelly, and rocks get pulled by gravity, and then that's going to trigger hairs, and we're going to interpret that as I'm looking down, I'm looking up. And I can interpret all that with my eyes closed because I am that fantastic, right? But any of you can do that. So let's look at all the structures that we have in here. Again, we have, I'm going to go to this picture here. We have the bony stuff that we see. I don't think I have a model here sitting around anymore. I took it back. So here is the bony stuff that we see. We see the cochlea, here's the whole vestibule region here, and then we see our semicircular canals. And that stuff that we see on the outside, that's the bony part. That is the bony labyrinth, all right? The bony labyrinth, it doesn't do anything. Well, I mean, it does. It doesn't help us interpret the movements, but it's protect. It's bony material, so it's there to protect. What does it protect? It protects this membranous duct right there. Labyrinth, maze. So I guess this was a maze to the first people that saw and identified it. So it's this membranous tube that's inside that bony stuff that we see and identify on the outside. So that's going to be the membranous labyrinth. And as you can see, this whole sucker's connected. That's why really loud sounds can cause disorientation, right? Some dizziness, some vertigo, because you can see that membranous labyrinth right there is connected. A little bit of connection here between the cochlear duct, where we interpret sound, or where we perceive sound, I guess I should say. And over here with the vestibule and the semicircular canals. So really loud sounds can cause movement of fluids here that move all these hairs. So there's a lot of cool things going on in here. So we have bony labyrinth. Bony labyrinth surrounds the membranous labyrinth. Okay, protects. And this houses the stuff. Okay. So that membranous labyrinth houses all of our receptors. Between the two, and by between the two, I mean right here. Let's get a different color. Yellow work? Let's see. Right there. And right there. In the space between the bone and the membranous, okay, all right there, that is filled with perilymph. Okay, perilymph. It's between the two, between the bony and the membranous. Inside here, this is filled with endolymph. Endo means within. So the endolymph is what fills the membranous labyrinth. Okay. The perilymph is around it, around the membranous labyrinth. Okay. There is some uniqueness here. This is our exception to the rule, as we'll see next week. Okay, this has an unusually high amount of potassium. So potassium is what causes the depolarization, which is different from literally everything else that we've looked at. Sodium is always a depolarizer. Potassium is always a repolarizer or a hyperpolarizer. This is the one exception to the rule. Endolymph has a very high concentration of potassium, and it's actually the depolarizer here. Okay. This is our exception to that rule. Okay, this is our exception to the rule. And we'll see that especially when we start looking at sound. Okay, it'll be much more evident there. So here is some of these sensory hair cells that we have. Okay, they're referred to as stereocilia. Okay, they're not true cilia. They're not, whip-like motion. So that's why the book, goes into this whole diatribe about them not being true cilia, because there's no whip-like motion. They only move when they're stimulated, right? But again, they're just modified, epithelial cells with these hair-like extensions, and they are stereocilia. Okay, so all of these dudes right here are stereocilia. Stereocilia. Okay. And we have one exceptionally very large cilium called kinocilium. When this one gets stimulated, right, this one is going to move one way or the other. And you can kind of see, down there, it depends on which way we're going to. Are we being bent towards that kinocilium? Are we being bent away from? And by that, I mean stereocilia. If the stereocilia move towards the kino, you can see that stimulates a heck of a lot of action potentials. When we move away, when the stereocilia move away from that kinocilium, it hyperpolarizes it. It hyperpolarizes it. As you are sitting there right now, not moving, not doing anything, we have potassium channels that are partially open. Okay. So these are going to be channels that we see here in this inner ear region to help us interpret where we are, movement, and sounds. Our potassium channels are always going to be partially open. All right. They're going to help when it comes to this interpretation here of movement. They're going to be partially depolarized. If I'm partially depolarized, again, calcium voltage gated channels are open, which causes neurotransmitters to ultimately be exocytosed and yada, 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 the same diatribe that we're going to be looking at 1,000 times here, okay? Same thing we saw with the synaptic knob at the end of the axon. Voltage got close to the synaptic knob, triggered calcium voltage gated channels to open, calcium rushed in, causing the vesicles to move, and same thing here. Okay? Same thing here, it's just my depolarizer is potassium. One of the most common things missed on the exam is this one stupid thing. It's the one exception, okay? It's the one exception. I don't know if that's a hint or not. I don't know what to tell you, okay? So when the stereocilia go toward the kino, that channel completely opens. Potassium comes rushing into the cell. I cause even more calcium voltage gated channels to open up. So I have more calcium comes in. When more calcium comes in, I can get more vesicle movement, more V-snare, T-snare interaction. I can dump out more, I can exocytose more neurotransmitters. The opposite. If we are bending away from the kinocilium, and here's the kinocilium right here, okay, and I'm bending away from it, that will close those potassium channels, causing the closure of calcium voltage gated channels. And then I get no release of neurotransmitters. The book gives a very simplistic look about it. I believe over in the interpretation of sound, I have a much more detailed kind of picture of it. All right. But to interpret body position, okay, when those stereocilia move towards the kinocilium, that's going to cause those potassium channels to open up completely, right? In their resting state, they're partially closed or partially open, however you want to visualize it. So I have some degree of depolarization. Again, this allows me to interpret I am standing right here. I am standing here. I am upright. I'm not looking left. I'm not looking right. I'm looking straight ahead, even though I'm not moving, okay? But this potassium channel being partially open allows for some depolarization to occur, okay? Which means I can get some calcium to come on in. I can get some release of neurotransmitter. Now, once I move and I bend towards that kinocilium, okay, here's my kinocilium here, okay, get over here in the field of view, and I bend towards it, all right, that's going to completely open these suckers. Completely depolarizes it. All of those potassium channels are open up. Excuse me. Allows a hell of a lot of calcium to come on in. More neurotransmitters can be exocytosed. And I can interpret that as, you know, I'm bending to the left or bending to the right or whatever the hell I'm doing. When I bend away from that kinocilium, I basically shut all of those potassium channels. And there's a number of proteins involved with that. Okay, and I'll show you the picture next week. When that bending away shuts all those potassium channels up, Again, they're mechanoreceptors. I'm mechanically moving. These are mechanoreceptors. So when I move away, that shuts them all up. And when I shut it up, now I have no calcium voltage gated channels being triggered to open. And I release no neurotransmitters. Okay. So now I can interpret that as whichever way I'm moving towards. Okay. What's that look like? I mean, right here. When I bend two, here's normal resting position. Okay, I get some depolarization. So I get some degree of action potentials to interpret where I am. As I start to move, okay, if I'm moving forward here on the example, here you can see I'm moving towards the kinocilium, the tall guy. And I increase my depolarizations. I've increased the neurotransmitters. I've increased the depolarization event. I go backwards, as the example here, I'm moving away, these stereocilia, I'm moving away from that kinocilium. And that helps to lessen the number of depolarizations that are occurring there. Okay. Same thing if I look down. If I look down, I'm going away from the kinocilium. If I do this, if I look up, I'm going towards the kinocilium and I'm going to increase my depolarizations. What if you get picked up and held upside down? Again, it's going to be a very kind of static thing, but there's a little bit else involved with that as well. Here is that otolithic membrane right there. There you can see that gelatinous goo. Okay. And then there's the little pebbles. And gravity pulls those pebbles. So when gravity starts moving, when you start moving, gravity exerts its force on you, and that helps to move these pebbles, which helps to move the jelly in certain different directions, okay? Which allows me to interpret if I'm going forward or backwards, or if I'm looking up or looking down. Again, you learn those body positions. Okay? But all that information is dictated by how many action potentials. Am I depolarizing it? Am I hyperpolarizing it? We have the semicircular canals and the utricle and sacculae involved with this static and dynamic equilibrium. Utricle and saccula is static equilibrium. Okay? Linear acceleration. The utricle and the sacculae are going to be involved with that. Okay. The utricle and sacculae have those otolithic membranes, within them. And as you can see, they're orientated in a slightly different way. One's more orientated in a vertical and one's not so vertical, more horizontalish. Okay. So when it comes to these two things, I can interpret whether or not I'm going up or down or side to side based upon the type of movement and which one gets stimulated. Again, if it's going to be that sacculae right there, okay, if I go up and down, that's the one that's going to be involved with that depolarization. If I'm going left or right, going more horizontal, then it's the utricle that's going to be being stimulated and sending impulses in. Okay, and I can interpret that as, okay, linear motion. Again, the stereocilia and the kinocilium. The stereocilium is embedded in a gelatinous otolithic membrane. They are otoliths. These are little stones, ear stones. Okay? And they are calcium carbonate crystals. That's all they are. That's all they are. And again, like I said in that picture earlier, gravity pulls those stones and that moves the gel. When I move the gel, I move those hair cells within them, the stereocilia. Again, if I move towards the tall guy, towards the kino, I further cause depolarizations. I increase it. Opens it completely up, greater depolarization, calcium rush in, yada, 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 release more neurotransmitter. More neurotransmitter, greater action potential, greater number of action potentials. Conversely, if the stereocilia move away from it, closes them up, and I inhibit it. I inhibit the release of action potentials. And again, it's going down, I assume you guys knew that, the vestibular branch of the vestibulocochlear nerve. Again, the utricle and the saculae is within the vestibule, right? Is within the vestibule. The last one we'll talk about here is dynamic equilibrium. Same hair cells, except now they're not in the maculae of the utricle and the sacula. They're in something else. Okay? They are in something else. Semicircular canals, like I mentioned earlier, one in each plane. So you can interpret X, Y, and Z movement. Each semicircular duct filled with endolymph. And at the base of that endolymph, okay, at the base of that duct, I'll just draw a duct. Okay. Okay. We have an expanded end. And that expanded end is called the ampulla. Okay. Let's put A for ampulla. It has hair cells. It has stereocilia. Okay. It has some hair cells. It has stereocilia extending up. Okay. Let's just go to a better picture. Okay. So here is my ampulla, my expanded end, okay, of the semicircular canal. There are my hair cells right there. And there's my stereocilia. My stereocilia are embedded in a cup of jelly. Not an otolithic membrane. Okay, that's more of this. Okay, I got otoliths, I got a gelatinous layer, and the otoliths exert a force down because gravity is pulling them down, right? Again, that helps me to interpret, all the stuff that we just talked about. Here, my hair cells are stuck in what's called a cupola. And the cupola is the gelatinous matrix here. Otolithic membrane in the sacula, in the vestibule, okay, the utricle and the sacula. And here we call it the cupola. Same thing. Here is my semicircular canal. Again, when I'm not in motion, okay, I have some opening up, so I have some depolarization, I have some action potentials. Whenever I move away from it, I hyperpolarize. Whenever I move to, I depolarize. So it's the same stuff that we just talked about. Okay? It's the same stuff that we just talked about as we just talked about with the utricle and the saculae. Except now, this force that's moving those hairs, or moving those cilia, stereocilia, is not coming from otoliths. It's not coming from little pebbles in your head. It's coming from that endolymph, from that fluid within it. That fluid is what pushes those stereocilia either towards it or away from it. So here you can see I got that wave of fluid moving. And as you can see, it causes those, well, yeah, okay, causes those hair cells to start moving away from it. So there you can see I start to depolarize it. I close it up. Okay, hyperpolarize it. Here, when I move in the other direction, fluid comes this way, and you can see the force that gets exerted there, and it helps to push those stereocilia towards the kino. And again, that opens them up a heck of a lot more. And I increase my rate of firing. Okay. So now we can interpret whether or not we're moving left or right. Okay. We're accelerating to the left, if we're accelerating to the right, by how those action potentials are being sent to the brain. Keep going around and around and around and around in circles, and now you're going to start, this is why you're walking around all wonky when someone spins you around and around and around, because you have waves going this way and then waves coming back through the other side and waves coming back and waves going backwards. Because as soon as it hits that cupola there, boom, the water is going to, or the fluid, the endolymph, is going to start going back and rebounding the other way. And then rebounding, and then it keeps going until that wave of fluid movement within there stops bending the hairs. So now you're not going around in a circle and feel like you're going to yak all over yourself. We have a vestibulocochlear nerve that sends all this information, is the vestibular branch of the vestibulocochlear nerve specifically. So we have that vestibular apparatus comes in and sends information into the brainstem, specifically the medulla. Okay. I'll just put MO for medulla oblongata. Okay. We have proprioceptors doing the same thing. Okay. If you're turning around and looking around and going really fast side to side, not only am I going to throw up here, okay, not only is the fluid within my inner ear moving, but also I have mechanoreceptors here in my neck. You have a ton of them here within your neck. All right. That is sending in information there to that medulla oblongata, into that vestibular nucleus. Now, if I open my eyes and I bring in the visual field, oh my God, now I'm converging a ton of stuff here, right? Cerebellum gets in on the act, right? With learned coordinated movements. Okay. So if you're going around and around and around on a tilt-a-whirl, I mean, the cerebellum knows that you're going around in a circle and you're going really fast. So all this information comes in, helps to control your eye movements. So you can try to focus on something. Control body movement to get you to stop going. So if you're on a tilt-a-whirl and round and round and round and you get off, the vestibular nuclei is bringing in all that proprioceptive input, bringing in still that vestibular apparatus. Not as much information coming in from the proprioceptors now. And it's sending down information to control how you're moving, because the vestibule is still being stimulated in some sort of way. So we can control all these movements. So everything here is connected. Learned body movements, inner ear, eyeballs, proprioceptive input. All of this goes in to affect how we move and where we focus our eyes so we can move properly. When someone's been on a tilt-a-whirl, a lot of times you look at their eyes, right? And you see their eyes are still, they're still moving around. Okay? Because there's information coming in here. Vestibular apparatus, the vestibule is still sending in information. Fluid still moving in that inner ear. So it's still sending in information. The vestibule sends out to not only to the body to move it, but also to the eye muscles, to the ocular motor, cranial nerve 3, to try to get control so you can focus on something on the horizon and right your ship and not be, moving all over the place. with it. All right. But that's why it's so damn hard to right that ship after you get off the tilt-a-whirl, because this thing is still sending in input. Okay. And you know that if you are looking in this direction, you know that you're going to want to start to move in that direction. And if you have a lot of information coming in, then you start going way too much in that direction. And then you fall over. And then you laugh. And then you vomit. Nystagmus and vertigo are a product of this movement that is occurring there. Again, it's triggered by that vestibular nucleus within the medulla oblongata that goes into synapses on cranial nerve 3. You can get rocks, those otoliths, they can become detached a lot of times with head injuries, old age as well. And they can become dislodged from that gelatinous material that they're embedded within. And they can start to move through that semicircular canal and cause some vertigo issues, okay, which can lead to some nystagmus when they turn and look in a direction there. And for the life of me, I've been sitting here trying to figure it out for the past 5 minutes. I cannot figure out. There is a test that you can do on an individual to determine whether or not their vertigo is based upon medication, something else, a virus, or if it's based on one of these tiny little pebbles getting dislodged. And you look for nystagmus. You have the patient sit down on a table and you bring them back really fast. And you're holding their head. Okay, you got a hand behind their back and a hand behind their head, and you quickly bring them down. And you watch their eyeballs. You turn their head to the left. You do it. You have them turn their head to the left or right. Now, they would have told you looking to the left is what makes me feel like I'm on a tilt-a-whirl. Okay, so you bring them back really fast. Okay, to trigger that fluid movement, right? Move that. Turn them. Either I go away from what they told me and look at their eyes, and their eyes will start to move. If they don't have nystagmus, if they don't have the eye movement going in every direction when you turn, perfectly fine. Bring them back to center. Okay, move them in the other direction. All right. If that direction kind of does it, okay, bring them back up, reset, bring them back down really fast again, and move them in that direction. Then you'll just watch their eyes, and then they'll get really sick sometimes. But watch their eyes. Okay. Then there is a technique that you can do to help get that little otolith back into where it's supposed to be. Okay? To get it out of the semicircular canals, get it back down in that vestibule structure. And it is a cool as hell type of maneuver that you can do for that. And the name escapes me off the top of my head. I've had a number of patients that I've done that to. And it helps to alleviate it if that's the problem. If that's not their issue, if it's not a stone that's been dislodged, from the otolithic membrane, then I mean, doing that, it's not going to do a hill of beans. Okay, but you can see that nystagmus and how that goes hand in hand with the inability to decipher where you're at in the world with vertigo. So those two things are hand in hand because of that little nuclei right there at the medulla oblongata. It synapses in the ocular motor center, which goes and supplies some of the movements for the eye.